Hello, my name is Nur Kasanjayana and I'm going to share you guys about the one, which is the basic concept of statistical and derivations. For introduction, a normal distribution is known as a continuous symmetric shape distribution of a variable. There won't be any variables that precisely suit the normal distribution because it is merely a theoretical distribution. For a normal distribution, some of the often used parameters include the variance, standard deviation, and the mean. The mean is the average of the data or central tendency. Next, the variance, represented by the symbol sigma square, measures the separation between each scatter number in the data set and the mean. It does this by representing the statistical distribution about the mean among the numbers in a data collection. The standard deviation, on the other hand, is the variance square root and aids in assessing how consistently the data value changes over time. There are three objectives in this experiment. The first one is to calculate and describe the following descriptive statistics: mean, variance, and standard deviation. The second one is to understand the concept of correlation. The third is to determine the relationship between the two variables. Next, I will show you guys about the methodology. The first one is 10 leaves were taken and their length and width were measured and recorded. The mean, variance, and the standard deviation of the length and width of the 10 leaves were calculated. The data was tabulated on a graph paper. The second one, another set of 10 leaves was taken and the same process was repeated. This was done for 3 more sets of the leaves. The results were tabulated as a data matrix for set 1, set 2, and set 3. The mean, variance, and standard deviation for the width, length, and the width of the leaves were calculated. Therefore, the data from all 30 leaves was used to plot a graph of leaf length versus leaf width on a graph paper. Next, it was determined whether there was a relationship between the length and width of the leaves. The type of relationship, whether linear, which is straight line, curve, or other, was discussed. These are the results for group 1, group 2, group 3, and group 4. Next, for discussion, this experiment 40 leaves were randomly selected in terms of both length and width and were separated into 10 groups. The correct information about each leaf measurements were taken of its length, width, and range. Then the length and width of the leaves were plotted on a graph. Statistics like the mean, variance, and standard deviation were computed for every collection. One statistical technique, which was correlation, is used for figuring out whether there is a relationship between the variables. The associations between the length and width of the leaves was determined from the plotted graph. The graph demonstrates the linear relationship between the length and width of the leaves. The scatter plot shows the positive linear correlations, indicating that it is in this particular case, the length and width of the leaves were more proportional, that is, as the length of the leaves grows, so does their width. Thus, it can be said that both variables depend on one another. For question 1, the answer is the differences or similarities in the mean length of leaves among differences indicate variations in growth patterns or genetic traits within plant populations. These differences could arise from environmental factors such as light intensity, soil nutrients, or water availability, influencing leaf size across different species or within the same species under varying conditions. Similarities in mean length may suggest common adaptations to specific habitats or evolutionary process, highlighting how plants optimize leaf morphology for survival and function in their respective environments. Differences or similarities in the mean width of leaves among different sets indicates variations in structural development influenced by genetic factors or environmental conditions such as light exposure or nutrient availability. Similar mean width may suggest common adaptive strategies within species or populations facing similar environmental pressures, contrasting with distinct growth responses to varying environmental factors or genetic variability, illustrating how leaves adjust their size to optimize photosynthesis. Question 2. The answer is variance measures how much the measurements of width and length of leaves vary from their average values. High variance indicates the significant differences among leaf sizes, while low variance means measurements are more consistent. Based on this statement, the leaves have different measurements of width and length. It implies the variability in the dimensions. Variance and standard deviation quantify this variability, which is high values indicate diverse leaf sizes, while low values suggest more uniform measurements. Standard deviation is the square root of variance. It measures the standard deviations of the average and individual leaf measurements. It is easier to read because it is in the same units as the measurements. Next, the answer to this posted question is, it is necessary to have different sets of measurements for the measurements of leaf width and length because it can help in accuracy and precision of the measurements. It is also necessary for capturing variability because leaves vary in shape and size measurements, and to capture this variability, it is crucial for understanding adaptations and genetic diversity. So, in conclusion, the experiment successfully applied descriptive statistics, which is mean, variance, and standard deviations, to quantify and describe the measurements of leaf length and width. It utilized correlation analysis to determine that there is a positive linear relationship between these variables. This relationship indicates that as leaf length increases, so does leaf width, highlighting a proportional dependency between the two dimensions in the leaf study. This understanding contributes to broader insights into leaf morphology and its ecological implications.
Hello, my name is Noradina Atira, and now I would like to present about the lab 2, which is the factor that affect the distribution and abundance of organisms. Now, for the introduction. All living organisms present in an environment are considered its biotic factor. A population is defined as a collection of the individual belonging to the same species that live a shared habitat. A community is formed when the various species live together in a common area and engage in interaction. One method to investigate how organisms interact in the community involves to conducting a species inventory and analyzing the population size of each species in the area. This experiment showed the basic idea related to quadrates and a biotic element present to the study location and categorize the plant species found there. There is 4 objectives for this experiment. First, to introduce the simple concept of the quadrate. Second, to enhance of the power of the observation. Third, for the record the difference in the plant distribution within the quadrate. And the last one, to infer from natural observation the possible factor that affect the distribution and abundance of the organism. This is the methodology for this experiment. This is the result for the group 1, group 2, group 3, and group 4. Now, for the discussion. From this experiment, we already got a result of the population for several plant species that were inside the quadrate. As the result for the quadrate 1, the most found species in this quadrate is a species B, which is 5, and it has a higher density, which is 29.41. The less found species is the species C, D, and F, which is only one species in the quadrate, and its density is lowest, which is 5.88. Where the density, we calculate by dividing the total number of the individual of the species in the quadrate with a total number of the individuals. The abiotic factor that may contribute in this species is physical abiotic species, which is a soil. Soil type determines the type of plant of the species B that can grow. In quadrate 2, species A and B are the most abundant species, which is 3, with a higher density 37.5. The species C is the less common species with only 2 individuals found in a quadrate 2 and a density is 25. The slope is a abiotic factor that can be the reason for the species A and B very suitable to breed. Next, for the quadrate 3, species B, D, F, and G are the dominant species with the maximum density of 18.2, making up 2 individuals in total. Species A, C, and E is the rarest species with just 1 individual discovered in quadrate 2, and the density is 9.1. The abiotic factor of the 1 can influence the seed dispersal that affecting species A, C, and E distribute. Lastly, in a quadrate 4, species B in the most abundant species reaching maximum density of 29.4 and totaling 5 individuals. Species A and H are the least common species, found only in a quadrate 4 and 1 individual and a density of 5.5. The chemical factor and abiotic element might influence the growth of the species B by impacting the sunlight available to their survival. Now for a post-lab question. For the question 1, the answer is quadrate is a square or rectangle area utilized in ecology and biology to designate a set area for examining the distribution of an item across a vast area. Quadrates are commonly utilized for studying vegetation and monitoring the abundance and distribution of the organism across the diversity ecosystem. For the question 2, the answer is the 2 basic requirements of all quadrate techniques are random sampling and the size of the quadrate. For the answer for the question 3, the advantage of using larger quadrate are the larger quadrate can cover more area and is providing more accurate data, while its disadvantage are its larger amount of data can be more difficult to handle and analyze. For the small quadrate, the advantage are easier to set up the sample and allow more precise, detailed observation within a small area, while its advantage is may miss species and variation, leading to less accurate result. For the answer for the question 4, the ecologists face a challenge with a restrict entry and challenge in observing and tallying organisms. Moreover, collecting comprehensive data on the various distribution of the species is a lengthy and resource-intensive process due to need for extensive sampling. In conclusion, the distribution and density of plant species in the quadrate were influenced by various abiotic factors, helping infer natural factors affecting organism distribution and abundance. Species B was most abundant, likely due to favorable soil and chemical conditions. Soil type was a crucial in a quadrate 1, slope favored species A and B in quadrate 2, wind affect seed dispersal in quadrate 3, and chemical factor impact the sunlight's benefit species B in quadrate 4. This finding highlight the role of physical and chemical abiotic factors in shaping plants community and explain difference in the plants distribution within each quadrate.
Assalamualaikum, my name is Ainur Nabila Syafiqah binti Abdul Aziz. My ID number is 2023899948. I will explain about Chapter 10, Community Ecology. What is predation? Predation is the basic mechanism that controls the flow of energy through the ecosystem from the initial foraging of herbivores on primary process through the apex predators. Next, we have mechanism of pan-herbivore system. It contains of defoliation and consumption of fruit or seeds. So, how defoliation occur? Defoliation happens by destruction of plant tissues such as leaf, bark, stem, sap, and roots. Next, feeding on seeds can be damaging, although it facilitates their dissemination of seed primarily through phases. What is plant response after defoliation? Defoliation and subsequent growth alter the trees physiologically. Firstly, it can affect hormonal growth regulators that regulate bud dormancy. Secondly, it increases insect and disease attack. Next, it reduces reproduction resources for nutrient and mature cell loss. Unfortunately, defoliation can lead to death in coniferous tree due to lack of physiological traits for recovery. Next, we have predation fitness. Fitness of predator is the ability to capture the prey, while fitness of prey is the ability to elude or hide from predator. Effect on prey plant fitness. The effects of the predation are strong at the juvenile stages of plant development. Grazers concentrate on young leaves and thus remove nutrient. There are a few effects on herbivore fitness. Firstly, herbivore biomass consumption and reproduction are correlated in primary production. For herbivore, the quality of food is low, but not quantity, which is high in number. Next, we have low quality food such as tough, woody, fibrous, and indigestible. Next, we have high quality, young, soft, and green or storage organs. Food of herbivores is low quality and also hindered by plant defenses that makes food unavailable, hard to digest, unpalatable, and toxic. Let's move on to prey defenses. Firstly, we have chemical defenses. This occurs to prevent predators from eating them. Firstly, we have chemical defense that occurs in three situations. Firstly, by secretion to repel predators. For example, bombardier beetles are those that deploy explosions to defend themselves. With an audible pop, these beetles spray a concoction of boiling, irritating chemicals at predators that get too close. Next, we have warning coloration. Poison dart frogs, for example, come in bright colors such as red, blue, yellow, or orange. These colors warn other animals to stay away from the frogs, which secrete a toxic substance. Last but not least, for mimicry. In this form of mimicry, a deadly prey mimics the warning signs of a less dangerous species. A good example involves the milk coral and false coral snakes. Both the harmless milk snakes and the deadly coral snake mimic the warning signs of the moderately venomous false coral snake.
Is cryptic coloration. Concealing coloration or protective coloration will allow prey to hide from predator due to the similarities that involve pattern, shape, posture, movement, and behavior that make the prey less visible, so it can increase the chance of survival. It's commonly among fish, reptiles, and ground nesting birds. And as you can see, we can see clearly from the example that I gave, where is the animals. So the red one actually is the seahorse, and the second one is actually a species of lizard that looks like dried leaf. Next is flashing coloration. The animal who has these types of defense will have two colors on their body, which is the bright side and the dull side. So it can help the animals to hide from the predators when they was at rest, but when they move, they will expose the bright color. So as you can see from the example that I gave you, when the butterfly was at rest, it will expose the black color, and when they will start to move, to flying, so they will expose to us the color of blue of the body. We will talk about armor and weapon. Clams, armadillos, turtle, beetle withdraw in armor or shell. Porcupines, hedgehogs, echidnas has modified hairs, which is we call as quills, that was sharp and painful to use weapon when they was rigid. So when they was attacked, they will spread their quills to the predators. Or the tortoise, the turtle will let the shell to prevent the predators to eat them.
Hi, my name is Izora Espaiza Butinasi. Today I want to talk about cannibalism. The definition of cannibalism is killing or consumption of either all or part of the individual that is from the same species. There are two types of cannibalism. The first one is terrestrial cannibal, which are 50% herbivorous. And the second one is aquatic or marine cannibal, which are predaceous. Cannibalism is common in animal kingdom and even in humans. 50% of terrestrial cannibal are herbivorous, most appropriate to encounter shortage of protein, for example in snail. Meanwhile, in aquatic or marine habitats, cannibalistic species are predaceous. Cannibalism is found in stressed populations such as rabbit, sea lion, and hippo. Lion and tiger are the example of cannibalism that happen due to facing starvation or hunger. An animal for crowded or dense population even when food is available is hippo. What is the potential of cannibal? Usually older and larger individuals. In some species of organism, the young may be cannibalized or consume older and bigger individuals. Why? Cannibalism is a mechanism to regulate or control their population that reduce in specific same species, which are same species competition as food gets scarce. How long cannibalism will happen? Usually short term, so no chance of extinction of species. Cannibalism promotes selective fitness advantage, increases longevity, eliminates potential interspecific competition, and provides a meal. That's all from me. Thank you.
So today I'm going to present about the mutualism. Mutualism is a type of relationship between the host and the symbionts, where both organisms benefits and no one is harmed. Mutualism divides into two, which is obligate and facultative. Obligate mutualism is the situation where one organism cannot survive with the absence of other organism, while facultative mutualism is the situation where one organism can survive with the absence of the other organisms. So let's take a look for the example. The first example, which is obligate mutualism, I give an example about the relationship between fig tree and wasps. The wasps lay its eggs and also pollinate the figs inflorescence. On the other hand, the figs not only provide shelter, which is in their fixed fruits, for oviposition to wave, but also allows its larvae to feed on seeds. The second example, which is facultative mutualism, I choose the relationship between fig tree and frugivorous bats. The fig tree is a necessary staple in the Amazon fruit bat survival. Without this tree, the fruit bat's food source would quickly be depleted. Fruit bats return the favor to the fig tree by doing their own share of the work. That's all from us. Thank you. Bye.